Well, good afternoon. Thank you. Good afternoon. On behalf of Karen and Mary Lou, we're glad that you could join us. You all obviously we share a common interest in Western aspen, so this should be hopefully enjoyable. This is a pretty remarkable species. You've all seen versions of this range map. Aspen has a huge geographic range. I mean, it's from coast to coast, from essentially the Arctic Circle to Mexico. And when you combine the range of Tremuloides with very closely related aspen species in Europe and Asia, it's a global distribution. We're going to be focusing on Western aspen, so the red polygon here is sort of our area of concern today, or focus today. One of the things that I'll point out, and Karen may refer to this later, but the blue boundary on this polygon on this map is the extent of continental ice during the last ice age. And so the vast majority of the range of Western aspen in North America is coincident with land that was not available until not too many thousands of years ago. That's unlike Western aspen in the Rocky Mountains and the Sierras. Within our area, I mean, beyond just the broad geographic range of aspen, I mean, a truly remarkable thing about our aspen is its ecological amplitude. In the Central Rockies, aspen is associated with every major vegetation type between lower and upper tree line. You see there in the lower left a small aspen stand out in the shrub steppe. That's Artemisia. And it's associated up near upper tree line with Engelmann spruce and subalpine fir as well. My task here in the first part of this webinar is to review with you some of the things that we were, until very few years ago, we were absolutely convinced that we knew pretty unequivocally about aspen, and that includes the things you see here on the list: its relative tolerance, its regeneration ecology, the genetic diversity or lack of it, and its role in stand dynamic succession. And then following from all of that, I mean, silviculturists, we were pretty sure that we knew exactly how to deal with it, and it was one of the easier species to deal with. And in fact, that's still true, but the story is much more. There's a richer silvicultural story that we're becoming aware of. Well, first of all, now I'm dealing with conventional wisdom, or at least the conventional wisdom until a few years ago, and I'm going to take some liberty. I mean, I'm oversimplifying a little bit for the sake of the narrative, but our view of the relative tolerance of aspen is that it's exceedingly intolerant, not being able to make it in the understory. In contrast, for example, with its common associate subalpine fir, Baker, in his classic textbook, Principles of Silviculture in 1950, in fact, characterized it as very intolerant. There wasn't anything in his experience that any tree species in the United States that was more intolerant than aspen. We also were pretty much convinced that aspen was a creature of dependent on high severity disturbance. There's a clear felling on the left there, pretty traditional way to deal with aspen and to regenerate it. The image on the right is intended to be an exception that kind of speaks to the rule. This is a fire-scarred aspen. It survived a surface fire. And it certainly, when I came across this in the book cliffs here in Utah, it got my attention, and it took me a while to convince myself that indeed it was fire scarred. The aspen regenerates very effectively vegetatively from root suckers, so shoots arising from dormant buds on rhizome-like lateral roots. And we, over decades, we had convinced ourselves that, in fact, in the West, it's all about suckers, and that seedlings, seeding events, if they occurred at all in nature, were exceedingly rare and, in fact, ecologically and silviculturally irrelevant. Some of us went so far as to state that in print. This is from that quote of myself is from the regional silviculture text in 1995. We're almost certainly our failure to recognize seedlings in nature was to some extent, I'm sure, a self-fulfilling prophecy. The assumption was that they were very, very rare, exceedingly rare, and so the standard of proof that was associated with saying that a small aspen was in fact a seedling and not a sucker became prohibitively high. The bar was set so high that I'm sure that we were ignoring aspen seedlings that were right in front of us, just because we assumed that they couldn't be seedlings. Because of the nature of the regeneration and the dependence on high severity disturbance, another element of the conventional wisdom is that aspen is almost entirely associated with very simple stand structures. So nearly pure aspen stands with a single cohort. So even aged stands. So that's the common model. Another thing, I've lectured a lot of students and relatives and so on about the clonality in aspen, and not to put too fine a point on it, the story would go that aspen clones were easily identified from a distance. I mean, like from a watershed away, based on phenology and gross morphology and that sort of thing, and that looking at a landscape like this, you could stands were easily identified, and in fact, stands equal clones and clones equal stands for almost entirely. So looking until a few years ago, I would have looked at a scene like this and said, okay, I can identify four different clones, and make life pretty simple. The silviculture, the silviculture systems that folks would think about, because of all this, our understanding of aspen. In silviculture speak, we're talking simple coppice, so clear felling an area and relying on abundant natural regeneration from suckers, and that the stands arising from that, basically single species, single cohort, and with high forest, with species that arise from sexual reproduction from seedlings, silviculturists are concerned about genetic diversity and what a particular harvest system or silvicultural system might do to genetic diversity. With aspen, we just didn't worry about that. Genetic diversity was moot. And we're going to turn. So that's the conventional wisdom, and it turns out that probably like a lot of things in life, conventional wisdom isn't always wisdom in retrospect. So we're going to turn this over to Mary Lou.
Hi, everybody. Well, it's not surprising with the long-term drought that we've had throughout the West and the millions of acres of wildfire that we were bound to see some aspen seedlings established. The trick probably, though, was recognizing them on the ground floor. As you see in this picture, they can look like a ground cover. We see the mature aspen over on that bottom left-hand, one that fell out of a mature aspen tree, versus the lanceolate leaves that you see here on the seedlings. The reason why I was able to identify them as a plant pathologist is because I recognized the foliar disease on the small seedlings. You can see the blackened areas from shepherd's crook. Of course, aspen seedlings were identified from the Tetons and the Yellowstone fires back in the 80s from Kay as well as Rami and others. And they recognized both seedlings, small seedlings on the ground, as well as aspen in new areas several years after the fire where aspen had not been reported before. In Arizona, where I reside, there are several fires from which I and other colleagues have found aspen seedlings following fire, either because they established in new areas or the seedlings were found and growing right along suckers as well. One of the three major components that you need, of course, is the bare mineral soil conditions created by fire. In areas where there's runoff, as any other plant, they're not going to get established too well, but in areas where there's some flatter ground or some mulch or protection provided, that's where we're going to find these seedlings. The second thing you need, of course, is seed production, and we find it to be quite abundant, especially the year after fire. One comment that I wanted to make is, pollen is flying through the air from aspen in May, and seeds mature in early June, where I come from. But our fires occur from May through early July, and so you're not going to really get seedlings established the year of the fire because they would be getting burned up or conditions just wouldn't be suitable if it's going to be fire. But it's the year after fire that you start to get those conditions because you have the bare mineral soil, you have a lot of seed produced. The other and so here this picture just shows that we had some bare mineral soil. We got seedlings were able to get their roots in the ground. You can even see cotyledon leaves on the one in the center lower left, little tiny leaves. And then you see the mulch has been laid on the ground, which is going to help provide some moisture retention. Okay, the other thing I wanted to discuss was the moisture availability, and this just shows the climate in Flagstaff from 1950 through 2007. Notice that there's 4 seasons represented here, and there's different scales for the amount of precipitation that we get on average and how it has varied over time. What I want you to notice is the summer, because of our monsoons, there's relatively consistent moisture provided from our monsoon season, and even in the case of a severe drought that started in 1996. You'll notice for the winter and spring that we had persistently below average precipitation, but yet summer was nearly average during that same time period, which is going to come into play for whether or not aspen seedlings can get established even during severe drought conditions. Okay, so is this a seedling or a sprout? Well, of course, the only way to test on some little plant like this is the root system. And for suckers, what you see is there's almost like an albino type of stem below the ground. There's no change really in the diameter of that stem as it goes down to that where it connected with the lateral root, which you can see on that right-hand side photo. Versus seedlings, which are going to have a tapering root. They're going to have fine roots starting to develop on the sides. You can see that first year there's not much, and then the second year on the right you even get some lateral root development. on a project Karen Mock and I worked on. This is a 7-year seedling. She had me dig 2 of them out of the ground to make sure that they indeed did have tap roots. You can see there's a very strong lateral root that I dug up out of the ground that was already present. And here is that tree before it was dug out of the ground. The root system was pretty much directly underneath that log on the ground. These were found in a ponderosa pine plantation following fire. There had been no report whatsoever of aspen existing in this stand before the fire. There is some aspen up above on that hillside in the background. So there were 18 ponderosa pine plantations. They all had to be put in exclosures because of the great browse pressure in this area. So there were 4 areas, 18 plantations total. And what I wanted you to notice here was that upper right group of plantations had the most aspen seedlings. Each one of those green dots represent aspen stems that were sampled genetically to show that there were differences, genetic differences, in that they were seedlings. Here's a picture just showing the variation in bud break. These pictures were all taken the same day within the same fenced area, and just shows differences in those seedlings on when they were breaking bud. If there were suckers from a seedling, the bud break was identical. So we had, these are the 4 areas at the top of the plantations, and then the number in parentheses is how many of those plantations had seedlings, and then the total number of seedlings that we found. And then notice at the bottom the number of aspen sprouts, so that by 11 years after fire, there was a total of 316 stems, yet just 70 genets, or original seedlings. And then the star was to remind me to tell you that those 2 aspen seedlings that I dug out of the ground to prove that they did have the taproot and they were seedlings. Apparently I had left some of the lateral root in the ground because both of them had produced suckers that were genetically identical. And because this was an open area and these seedlings were spread out, the GPS would take you right back to the same location to seedlings. Okay, we also aged our seedlings by counting the terminal bud scale scars, similar to the work of Kay and the Grand Tetons and Rami et al. In the Yellowstone fire areas. And our results were similar to theirs in that 90% of the seedlings got established within the first 3 years following fire. And we had seedlings established as far as 6 years out. For the Yellowstone fires, it was 8 years. The majority of our seedlings, 60%, established within that first year. In 2002, we had our most severe drought year, so we didn't expect to see much there. Kay had found that the 60% of his seedlings established that second year, and he noted that that was due to moisture availability, that second year was the better year. Okay, so in this photo of the ponderosa pine plantation, what I can see is 6 different seedlings, aspen seedlings. And it just indicates that in a frequent fire environment, such as these ponderosa pine areas, there's a potential for genetic diversity among the aspen if they get established, since bare mineral soil conditions are being provided more frequently. We did find just 2 aspen seedlings outside of the fences. other Forest Service workers had noted more were present originally. They piled logs that were on the ground around this one, and it does show signs of browse impacts, as does the other, but the logs keep getting piled higher to protect it. Due to the absence of aspen seedlings outside those fences, as well as the impacts that we see on suckers following fire, we believe that the browse pressure in some areas is probably greater impact than that from climate change at this point. And now we're going to turn it over to Karen.
Okay, hopefully I can be heard, and let me just go on to the first slide here, maybe. All right, so this is an interesting situation if we're not supposed to have seedlings as a major component or a major factor in aspen regeneration in the West, but yet we're seeing evidence of seedlings after disturbance and after fire. So one of the big questions really that comes up after that is whether or not these seedlings might actually persist. And the one way to look at that is to look at the spatial pattern and the distribution of clones. And in order to do that, because I'm a geneticist and I, of course, am familiar with these tools, I can use microsatellites to distinguish clonal boundaries, when in the past, clonal boundaries, as Jim said, are really distinguished by just visual characteristics and visual traits, maybe leaf shape, leaf color, bud burst timing, things like that. But with a set of genetic tools, one of which is called nuclear microsatellites, you can actually use these genetic tools to distinguish the clones precisely. So that's what I did. I'm not going to take you through too much about microsatellites. You'll probably be glad to know about that, although I'm more than happy to answer questions about it. So instead, I'll just jump straight to the results here. And I'm going to try this arrow thing. Oh, look at that. Ah, there we go. Okay. So this is a map of some results that came from a large area. This is about a township, which is at the top of Logan Canyon in northern Utah. And this is a study we did some number of years ago where we sampled 812 aspen stems just on a 50-meter grid. So we just threw down a 50-meter grid right on top of all the aspen stems that were out there to see where the boundaries were. And to our surprise, we detected 195 different clones here, which was not at all consistent with what our expectations were. And this begins to suggest that maybe seedlings are not only occurring and germinating, but that they're also persisting over time. Because if you're persisting over time, then you're going to end up with a greater genetic diversity, and genetic diversity that might be a little bit patchy like this. So this is consistent with the expectation that maybe seedlings aren't something we want to ignore ecologically. Okay, and so if you look at these symbols, some of these symbols are reused from polygon to polygon or stand to stand here, but within each of these polygons that you can see here, each stand, the different symbols represent different clones. And we're very, very careful with this not to include, not to count as a separate clone something that was different just by one small genetic difference. So we really were pretty conservative in identifying these. Another thing I want to point out is that we used one symbol across the whole picture here. These little small white dots, you can see one there, you can see one there, another one there. Those are symbols that represent clones that were only seen one time. And so the other message there is that when you sample on a 50-meter grid, most of the clones that we see, we only saw once or twice, which again is really different from what our expectations were. So we decided that we needed to look at another area just in case this was an odd area that we had chosen. And so we did the same sort of sampling strategy down on Fish Lake in central Utah. This is the home of the giant pando clone, which you can actually see here, these dark circles like this. These are paired not because you're losing your vision or you have a migraine right now, but because we sampled a large tree and a small tree, wondering if the overstory was the same genid as the understory in most cases. So that's what this picture is showing. Now, so our results from this study, or this plot, suggest that, yeah, we do see some big clones. And the famous pando clone, which is supposed to be the largest living organism on Earth, is certainly one genetic entity. There are two other really big clones. This is one right here. You can see black triangles. And there's another one, a great big one down here in a different stand. So there are some big clones. But the surprise to me was not so much the presence of a few big clones, but what was happening over here. Again, these little white circles, that doesn't mean that these are all the same clones. That means that these were clones that we only saw once or twice. And so, and we call these the onesie-twosies. So we had a lot of onesie-twosies in here. So over this whole study area then, we detected 61 different clones, which was really surprising. And once again, by the majority of clones we only saw once or twice. So this pattern did seem to hold up, and it has held up since with additional study areas. Okay, so let me go back a couple of slides. One thing I didn't tell you about our symbols here. We found another interesting pattern when we were doing this work, and that is that we began to see three versions of certain genetic loci when we only expected to see one or two. And that, without being too technical about it, that suggests that we might be seeing triploids, which are a version of a genotype, a version of a genetic individual that has 3 copies of its chromosomes instead of the usual 2. Okay. So what difference? And who would ever care about triploids? And I'd like to try to convince you that this might actually be part of the picture, along with the commonality of seedlings. First off, triploids, if they follow other plant species, triploids may be more vegetatively vigorous. So that might be something we care about. Another is that the triploid plants are expected to be sterile, or at least to have greatly reduced fertility. That might be something else we might want to pay attention to. And finally, triploid plants are expected to have bigger cell sizes, and that may well influence their physiology, and in particular their ability to move water. And so this is a question that we're looking at right now. Let me go back to these pictures. In these pictures, in these symbols, the dark symbols, all the gray and black symbols here, those are all triploids. Okay, and so you can see a pattern here, and this is something that has certainly held. The larger clones tend to be triploids. That doesn't mean that all the triploids are big. There are some small triploids around, but the big ones tend to be triploid clones. And if you look at this slide, remember this is the pando clone, same symbology here. So the pando clone is also apparently one of these triploids. And this is another thing I wanted to point out. We've also extended this work on triploidy to the entire range of aspen. And there are lots of interesting findings here. I don't have time to talk about them all, but with respect to triploidy, it does look like triploidy is something that you especially find in the West. And this is interesting to me because the proportions here of triploids versus diploids, those are proportions of genotypes without any regard to the size of the clone. So if we know that triploids make up, say, 30 or 40% of the genotypes, the clones that are out there, but at the same time we know that the larger clones tend to be triploid, that suggests that in terms of the numbers of trees out there, there might be a great majority in many of these places that are actually triploid. And that raises, I think, some very interesting kinds of questions. These pictures right here show the actual chromosome squashes. So this is a picture of a triploid aspen clone, and this is a picture of a diploid aspen clone. Here you can see that this is the continental divide, obviously. This is the same last glacial maximum that Jim talked about in his talk. All right, so a few conclusions then from the genetic data. First off, triploids are apparently common, maybe quite common in many of these western landscapes. Most of the very large clones that we see are triploid, but most of the clones are small, some of them very small, and the patterns suggest that seeding events are likely much more common than we once believed. And now I will turn it back over to Jim.
Well, another thing that has, with respect to Western aspen, that has at least changed for me, and again, I'm going to somewhat oversimplify the conventional wisdom, but for the sake of the story, but there clearly are multiple pathways of stand development for aspen. Now that's been recognized. I mean, you folks have referred to stable aspen, a stable aspen type versus a seral aspen type, and so on. But the story's richer than that. And for me, I mean, speaking for myself, part of it has been really ignoring what was right before my eyes because it didn't fit the model. High severity disturbance, essentially a pure stand, single species, single cohort. When in fact, on certainly any landscape in the Rockies, there are examples of that, lots of examples of that, but there are also lots of examples of aspen apparently being pretty happy in mixed species, mixed aged stands, even including 300-year-old old growth in Engelmann spruce, subalpine fir, and aspen, which is some of the stems being as old as anything in the stand. The bottom line for me as a silviculturist is that the argument that we need to broaden our view of aspen. That it would include the multiple pathways of stand development, a broader view of aspen restoration than we perhaps have had, and also I think that a lot of this new information really suggests a revisiting our views of appropriate reference models for restoration. Now, all of this is made timely. Now, certainly silviculturists and others are concerned about aspen. And in my part of the world, the central Rockies, the idea of conifer encroachment, perhaps the loss of clones due to successional displacement, particularly on marginal sites, the rapid decline of some stands and clones, with loss of the overstory without regeneration. Those are all concerns that lots of us deal with. One of the things I talked about simple coppice being the traditional model. Well, it's clear to me, at least, that there are lots of potentially situationally appropriate alternative silvicultural systems that silviculturists at least ought to consider. Everything from group selection to different kinds of reserve systems, but certainly aspen can be a major player in many forest types as part of a mixed species and even mixed age stand. Our understanding of aspen genetics and regeneration ecology is dynamic. It's changing. The last 5 years or so have there's just a lot of really solid contributions to the literature to support this. This particular this particular image is an interesting one. It's here in northern Utah on the Deseret Land and Livestock Ranch that some of you may be familiar with, but a number of stands and multiple clones. And this area was subjected to a late spring freeze, and that was pretty widespread. It occurred over a very large area and affected everything from Gamble Oak to aspen. Interestingly, and all of these trees re-leafed, but the ones that were particularly hard hit were the clones that leafed out early. One of the things that is certainly going to change in the coming years is the idea of genetic transfer. I mean, for a long time, foresters have been concerned about seed zones and seed transfer guidelines, that sort of thing, in moving genetic material around. We're paranoid about introducing off-site material that may not be adapted, and so on. And that concern and that history is reflected for lots of species with a great deal of information about how to appropriately move material around. Aspen's been ignored in that, and I think that must change. The idea, when we start to think about assisted migration, which people are already thinking about, that's clearly going to be necessary to get a better handle on adaptations and how material can be moved. So to kind of summarize, I think I started this with my version of some conventional wisdom, but clearly seeding events appear to be actually fairly common. There's real-time evidence of that Mary Lou talked about, and there's also the signature of seeding events in the genetic work that Karen referred to. Within stand, genetic diversity is clearly much greater than we thought not too long ago, and it's almost certainly ecologically and silviculturally relevant. The ecological roles and I'm referring to stand dynamics and succession, in fact, are varied. Aspen's a remarkable species with a huge ecological amplitude, and it's probably in hindsight it was pretty silly that some of us had such a narrow view of what it could do. Its ecological roles are not restricted to the conventional model. And then finally, I would argue that there's a broad range of silvicultural alternatives which can and in some cases I think should be considered, that go well beyond the traditional simple coppice. Thank you very much, and I think we turn it over to Rose to moderate questions.
Yeah, thank you very much, Jim, Karen, and Mary Lou. This is Rose Long again. We have plenty of time for questions, so keep them coming in the chat pod. We have a couple that people have presented over the course of the presentation. And we'll start with this question for Jim. This is Barry Johnston. He says, I believe that the conventional wisdom specified clear felling as the treatment of choice largely because of hormonal control over sprouting. Has this perspective changed? If so, what are the known controls over aspen sprouting?
From my perspective, no. I think Barry's his statement is correct, and I think that again, from my perspective, what has changed is that that clear felling and that hormonal, I mean, you had to decapitate all of the stems in order to get successful regeneration over fairly large areas. And I think that has changed. That it's able to regenerate vegetatively and from seedlings with much less heavy-handed disturbance. So that would be the change, I think, from my perspective, not the physiology of vegetative reproduction.
Good answer, Jim. This is Rose again. Here's a question for Karen. This is from a guest. The guest says, one of the ideas we're considering regarding climate change is treating high elevation mixed aspen to stimulate suckering, so there's more seed production in the future to colonize areas that become newly suitable as the climate changes. Do we need to consider whether these stands are diploid or triploid? Are there any field characteristics that can give a clue?
Wow, that's a great question. I wish we had two-way video so we could gesticulate a lot. But yeah, that's a really neat question. If you're looking at seed sources, then yeah, triploidy is something you really want to be cautious about, because we really have not known to ask questions about triploid until now. And the result of that is that we don't know very much about it. We don't know just exactly how fertile they are. We don't know how frequently they produce flowers. I know Pando produces the big male clone that produces flowers. We don't know if there is anything like pollen swamping. There's just a whole range of things. So if I'm understanding your question, yeah, we do need to be, if you're looking at seed sources, you definitely need to be looking to see if your sources are triploid, because you're not going to have very much viability. I don't know if that answers the question.
Field test.
Field test. We need a field test, Jim says.
No, I think that was part of the question. Is there a way to that you could when you walk into a stand, can you can you identify sort of without genetic magic?
No, you need genetic magic. We have, I've been fiddling with that out in the field, because we do have a lot of clones that have, or trees that have been tagged, and they have a known ploidy differences. And to tell you the truth, I'm seeing a whole range of phenotypes, and I'm not seeing anything that's really jumping out as a strong indicator, other than just flat out clonal size.
And we have another question here about diploids and triploids for you, Karen. This is from Wayne Shepard. He asks, do diploid aspens sucker as well as triploid genotypes?
Hi, Wayne. Thanks. That was a good question, too. That's a difficult question to look at, because aspen clones are so extremely variable that the variance among clones may completely swamp out the variance among triploids and diploids, if that makes sense. So that's kind of a hard thing to look at. However, I'll say that we have a collaboration going in Rick Lindroth's lab. I'm not sure if Rick is here today, but we are beginning to look at physiological differences between diploids and triploids, mostly associated with the economy of water. But we're certainly tracking the number of suckers, and that there may be something there. I think that'd be an interesting question to ask, but a really difficult one because of the background variance in clones, in root health, things like that. So I'd love to know that.
And we have a question here that maybe Mary Lou can answer. This is a question from Barry Johnston. He asks, how do single-species aspen stands naturally reproduce, and what about serial stands that have multiple aspen generations?
Yeah, I'm not sure that that we have the answer to that. One thing I was going to add to the conversation was that we found that just 60% of our seedlings were sprouting, so certainly it's going to affect some of the work that Karen has been doing in the more mature stands, that whether or not they seeded in earlier or not is going to depend on how well. You won't be able to tell just from testing, because you don't know how well they vegetatively reproduce. But certainly the large triploids are showing that they are vegetatively reproducing. in our early work with our young seedlings, the triploids aren't standing out right now as far as their vegetative reproduction.
Thanks a lot for that. Do you have a comment on that, Karen or Jim?
We could add a little bit here. I think it's interesting what Mary Lou is saying about the low number of triploids that we know about in that study. I have a graduate student, Richie Gardner, who has also done a little bit of work looking at the in trees that are 5-inch DBH or so, looking at the last, I believe it was 10 years of growth. And we were not able to find a difference between diploids and triploids with respect to increment in those years. But I do wonder if there might be some differences early on that they could be physiologically based, they could be hormonally based, I don't know. But I think looking at the early stand development in these triploids and diploids might be a really interesting question.
All right, we have a question here from Rob Taylor. He asks, would fire be contraindicated if a stand was a single triploid?
Jim is shaking his head, and I agree with that. In fact, it might kind of be the opposite. If triploids are the way to maintain a triploid in a landscape would be through coppicing or through fire or disturbances like that set it back and stimulate suckering. So, but that kind of management would certainly, I would think, would favor triploids, especially if the triploids are the big ones. And so you might have you might tend to favor triploidy by that kind of disturbance that causes lots of suckering, but at the same time you're potentially losing genetic diversity at the larger scale over time because you're going to be favoring the large suckers and the clones that can persist by suckering. That may be the triploids, and we might be losing genetic diversity, especially if we're not allowing some of these or recognizing and conserving some of these seeding events.
Here's an observation from Dave Tart and a question on the end of it. He says polyploidy is related to drought tolerance in shrubs. Could that be why triploidy is more prominent at the south end of Populus tremuloides range?
That's pretty, I mean, speculative, but it certainly makes sense to me. I mean, I think that when I first became aware of Karen's and her collaborators' work with the triploids and the fact that it's much more common in the Rockies than it is in the Boreal, for example. It seems like that's one obvious correlate. So I think it's a good hypothesis.
I'll add one thing to that, and that has to do with cell size. If you look at what we expect in triploids and diploids is that triploids are going to have larger cell sizes. What we're looking at right now, again in collaboration with Rick Lindroth's lab, is that there may, if that translates into xylem element size, then that could have a lot to do with how these triploids move water. So triploids may be better than diploids in certain climates, and maybe in past climates. But as we start seeing new levels of drought and maybe drought in new areas, things like that, it may be that triploids actually have a disadvantage because they may well cavitate more easily. And so that's a kind of question that I'm very intrigued by, and we're trying to set up common garden studies and even in field studies to begin to look at hydraulics in triploid versus diploid aspen. Again, we're certainly going to run into the problem where there is so much clonal variance in water-moving characteristics, water-moving traits that that may swamp out differences between diploids and triploids, meaning that it may require a ginormous sample size. So that's yet to be determined.
We have another question here. This is from Randy Fuller. So if you're going to start an assisted migration program, would you look to diploid or triploid?
Wow. Randy, that's, I'll offer one thought, and I have this fantasy that triploid clones, that they're going to be some superstars in terms of fiber production. And so if I had some kind of a fiber focus, that maybe I would be using triploids. That's somewhat speculative, but that's a possibility. On the other hand, if your goal is to conserve genetic diversity, I'm thinking that the focus would be on diploids, because they're going to. I mean, you've got the opportunity for further genetic diversity arising in the future.
Yeah, I would agree with that. And some of the answer to that question may come from how easily triploids are to propagate versus diploids. One of the things I did not mention is that in a particular seed crop of aspen that comes, from diploids, presumably, that your initial proportion of triploids is going to be very, very low. That's what we expect. So if we're seeing a high proportion of triploidy over time, that's very likely to be the result of site selection, of natural selection happening. And so if you are able to maintain genetic diversity and do seedling plantings to maintain aspen, at some point you will be regenerating and potentially selecting for triploids. So it probably depends upon your management goals and the time scale over which you're interested.
We have a couple of questions about how to distinguish sprouts from seed, and also how to distinguish diploids from triploids. This question is from Diane. She asks, she says, when we survey disturbances, we don't collect data in a way to distinguish sprouts from seedlings. Is this something we need to do in the future?
Yeah, I think Diane, you probably are really looking for more work. I'm shooting from the hip here, but I'm thinking that no, that isn't something that would be realistically and routinely included in your regeneration monitoring. Rather, I think with just an appreciation, an increased appreciation that seedlings might be present as opposed to just suckers, that in events where, especially in situations where because of distance from the rarity of aspen in the pre-disturbance stand, that kind of thing, where you would expect possibly more seedlings, that maybe an increased effort to protect those might be warranted, again, along the lines of conserving genetic diversity. So if you had a limited fencing budget, I mean, that might be one of your decision points about where you're going to spend money for fencing, that kind of thing.
And I was just going to add that part of the reason for doing this webinar was to get the word out that it is happening, so that there is an awareness. And if you should happen to find them in an area, especially where you're worried about impacts from browsers, as far as some of the areas we have where we've had a pressure for 100 years plus of heavy ungulate pressure, and that and on top of that there's a selection pressure going on for drought-resistant stems out there, that the awareness, and if you do happen to see them, that they can be protected. So not so much looking for them as doing something when you find them, I guess.
And Here's a question from Keith Gustafson. He says, my understanding is that to distinguish clones that are diploid or triploid can only occur in a lab using microscopes. Is it realistic to consider this characteristic when planning treatments to regenerate or maintain aspen?
Yeah, I can take that one. That's another really good question. It turns out that there are a couple of additional ways to identify triploids versus diploids. They're not as reliable as looking at chromosome squashes in the microscope, but that is incredibly labor intensive because you have to grow aspen up in the greenhouse, you have to get the soil just right, you have to pick off just the right root tip, and then you have to make a bunch of squashes until just the right one appears. Those slides that I showed of the microscope squashes of a diploid and triploid are the product of about a year or more of work. So that I would agree that looking at a microscope and counting chromosomes is not very practical. However, if you use microsatellites and begin to see 3 alleles per locus, that's a fairly quick and easy test. And that certainly could give you a very strong indication that it's a triploid. In addition, there's a technique that we use most of the time called flow cytometry, and it's a quick way to look at differences in nuclear density. It also is not as clean and not as certain as the microscope test, but it's a really, really good indicator, and we've been able to pick out diploids and triploids fairly reliably using one or especially both of those.
Here's another question. This is from PC Arborist. I hope I'm interpreting this question correctly. What would the percentages of diploid and triploid aspen be in response to climate change?
That's another good question. I wish I knew that. I'm looking around here for somebody to ask. But I think it depends on the relative differences that we find between diploids and triploids. We don't know if there are certain things that are universally true about triploids versus diploids that might give them an advantage or potentially a disadvantage with projected future climates. And so that's the kind of research that we're looking into right now, and there are a whole bunch of real exciting questions. And I know that's not a very satisfying answer, but it is an area of active research right now. One thing it occurs to me that I did not mention, I probably should have. I have a pair of undergrad students right now that are looking at a test that may help us distinguish diploids and triploids, and that's the size of the stomatal opening in the back of the leaf. You could actually, potentially, you could measure that using a field microscope, and if it's reliably different between diploids and triploids, that could be a nice field assay. We don't know the results of that yet, but we have a lot of leaves that we have taken microscopic images of, and we're analyzing that right now.